Hello, everybody. Okay, so now we made lots of copies of our DNA using PCR, and we can use it, and we use it in something called gel electrophoresis. So in this assessment statement, it wants us basically to state, so we got to remember that gel electrophoresis, the pieces, the fragments of DNA, they move an electric field, and they go different distances according to how big the DNA molecules are. Pretty vague. So let's talk about that real quick. So in our PCR, in our polymerase chain reaction, we end up making lots and lots of copies of DNA. So let's just look at just one piece of DNA, just so we can simplify it. So how do we break this DNA into different sizes? So the first thing we need to do is we need to expose the DNA to something called a restriction enzyme. So restriction enzymes look for certain sequences, and it depends on the type of restriction enzyme, but they look for a certain sequence of nucleotides. So let's say like this restriction enzyme is looking for the sequence AATT, something like that. On the other side of our DNA, right, because they're complementary base paired, it would be like this, right? C always binds with G. Yeah, that's supposed to be a G. G with C, A to T, blah, blah, blah. We're going to talk more about exactly how it cuts, but for simplicity, let's say in our sequence AATT, the enzyme looks for the space between the A and the T, and that's where it cuts. So AATT, I've identified it, and I'm going to cut between those two. And there's going to be lots of places in your DNA where is the combination of letters AATT, right? You have 3 billion bases in your genome. So let's say there's an AATT here, there's an AATT here, another one here. So when we're done, we have 4 different pieces of DNA. Our largest sample looks like it's 4. I wish I made these a little bit bigger. I'm just going to make this a little longer just so it fits to our argument. So our longest piece of DNA is 4. The next longest is 2. The next longest is 1. And then our shortest piece of DNA is DNA segment number 3. So we need to separate it. And we separate it in this machine called a gel electrophoresis. And a gel electrophoresis is just a box of jelly. And this jelly in here is like really hard to move through. It's like a sponge. It's called an Argos gel. And we have this Argos gel, and on one side of it, it's negative, and the other side, it's positive. And eventually, we're going to hook it up to a power supply. We also create little wells. So think of these as little holes. And what we're going to do is we're going to put different samples of DNA in here. So let's pretend that this is Bill's DNA. We found some mystery DNA, but we found Bill, and we're like, Bill, we need some of your blood because we think you're the murderer, and we want to see if your DNA matches the DNA at the crime scene. So we put all of Bill's DNA, well, at least from the sample, into this first well, into this first little hole, and then we turn the machine on. DNA is slightly negatively charged. And because it's negative, it's going to be attracted to the positive side. The pieces of DNA that can travel the fastest are the small ones because they're super agile. And they can go through this gel, this sponge, and they can be zip, 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 zip. And then we turn the machine off. So segment number 3 would be all the way down here because it's the smallest. Segment number 1, the next smallest piece, is like, I'm not super fast, but I was faster than those other pieces of DNA. It would stop there. Segment number 2, maybe it would stop right here. And then segment number 1, which was the biggest piece, traveled the least far. And then what we can do is we can look at where this stopped, and we can match it with an unknown. So maybe we found DNA at a crime scene that stopped at the same spots. And we could be like, Bill, you are going to jail because your DNA matches the DNA we found at the crime scene. And that's how you use gel electrophoresis.